The interpretation of FNIRS or FNIRS data can be difficult for novice users, as the data usually cannot be quantified. Our protocol using two complementary methods provides a better understanding of this data. The main advantage of this technique is the increase in the reliability of the data analysis using two qualitative GLM-based statistical parametric mapping and comparative hierarchical mixed models for multi-channels.
Before performing an FNIRS experiment, place the FNIRS device in a dark, noiseless area and place a whole head FNIRS recording cap onto the head of the subject such that the position corresponding to the center of the International 10 to 20 system is located at the number 245 holder of the head cap. Attach the marking sticker to reference location points and take pictures of the subject's head with the probe locations, including the reference points from 15 perspectives. Arrange the 48-channel system with 32 optodes to a head cap bilaterally over the frontal and parietal areas as regions of interest, and use the 3D digitizer software to determine the spatial registration. After scanning picture data for the entire head, determine the spatial coordinates of the subject by auto-measurement and save the data as origin and others files. To perform an FNIRS analysis, have the subject get into a comfortable position.